welcome to a new episode of the Computomics podcast. I'm delighted to talk to our guest today, which is Nadine Ziemert, the professor for translational genome mining at the University of Tübingen. Hi, Nadine, welcome.
Hi, nice to meet you.
You too. You know, we love to start with a little icebreaker here. And I was wondering, some people swear by honey or hot lemon or tea. What's your favorite home remedy, if you have one?
Sleep.
Sleep,
yes.
So wise. That's, yeah. I think I'll steal your answer if I'm ever asked that question again, because that's so true. Sleep helps with almost everything.
Yeah, at least that's what I was told by my husband, yes.
But your research obviously focuses on different remedies. You are looking for new antibiotics at your lab at the University of Tübingen. How are you going about this search?
Yes, I have to say, I don't know if we're really looking for new antibiotics because we do it kind of indirectly. Mm-hmm. So in the past, new antibiotics have been found by looking at bacteria, because actually I thought that's always quite interesting that the organisms the compounds fight are also the organisms where you can actually find the antibiotics, right? Mm-hmm. So most antibiotics are produced by bacteria to combat other bacteria as kind of a warfare or to have an advantage against other bacteria, you know. Mm-hmm. And so in the past, people have grown bacteria and then just looked what they produce. They have looked chemically, so they have extracted the bacterial cultures and the extracts, they checked if they kill other bacteria, for example. And we don't really do that. So we go a more indirect way. We look into the genomes of these bacteria because they're, within the genes, this is kind of the blueprint of what bacteria can actually produce. And there's a massive amount of genomes out there that we can easily just get from public repositories. So we just look in all kind of bacteria for the genes that are responsible for production of these antibiotics. And then we can computationally predict which bacteria can kind of make what kind of these antibiotics. And they don't just make only antibiotics, they also make other compounds. They have, for example, they make compounds that are active against cancer cells or that are anti-inflammatory or proteasome inhibitors, all kind of compounds. And we're really looking in the genomes for the genes that encode for these compounds.
Interesting. So if we were to expand the metaphor you introduced at the beginning, if the original way was to kind of scour the battlefield of bacteria for kind of the leftovers and what might work, or like weapons lying around, you actually are the spies looking into the blueprints kind of hidden away and trying to find how to attack the bacteria that way. Would that be a good way to express it?
Yeah, maybe. I'm not sure, but I think it sounds cool if you say it this way. Yeah. So, we know that these bacteria produce these interesting compounds or these antibiotic compounds only under very specific conditions. And so even bacteria that we grew in the lab for years and years as kind of our pets, especially known for producing antibiotics are the Streptomyces bacteria in the soil. So they were known to produce antibiotics, but maybe one strain was known to produce 3 of these antibiotics. But if we look in their genome, actually, we see that they can produce many, many more. So each one of these different Streptomyces strains can produce up to 25, 30 different compounds. And something we never see in the lab because they are highly regulated. They sometimes contain up to 7 different regulators. So you really need very specific conditions to see them made in the lab, and that is hard. But if we actually look in the genome, we can see the full potential of each of these strains. So we can really say who can produce a lot of these compounds. Then, of course, the next step is to see, can we also kind of predict when or how they produce these compounds. That is the next step that we also try to predict. But in general, we know now by looking at the genomes much more about how versatile or how diverse compounds these bacteria are actually able to produce.
I have two follow-ups on that. So one is how you said a lot of them only produce like maybe on average 3 compounds, but you know from looking at the genome that they have the potential to produce 30. How do you know that? Like, what about the genome tells you that? What do you compare it to?
Yeah, so there's specific signature domains that we can detect bioinformatically. So we know usually there's about 10 different classes of compounds that have interesting bioactivities that we're looking for. So these secondary metabolites or nature products, we call them. And there are certain signature domains that we can detect with rule-based methods that we know. And the different compounds that are made are actually mainly puzzles, Lego pieces, where these signature compounds are just put together in a different way. So if we know there is a certain signature domain with a certain other domains near each other. So in bacteria, it's really nicely because usually the genes that are responsible to make or to produce one compound, they're all near to each other. And these special nature products or secondary metabolite genes have a very specific signature that we can usually read and detect in the genome bioinformatically.
And you spoke about that second step. So it's also about finding out when do they actually, when and how can we get them to produce the full 30 compounds rather than just their 3 that they're maybe currently putting out. What have you found out about that? Like, what are the factors, or how do you build that environment maybe?
Yeah. For example, we can predict in a lot of cases if something is a siderophore. These are compounds that are usually produced under iron starvation. So the bacteria need iron, so they make these specific compounds that they are able to complex iron, and then that's really helpful for the cell. So, and there are certain signatures within the gene cluster that we can read. For example, they have a certain transporter in the gene cluster, they have a certain regulator, they have certain binding domains, transcription factor binding domains that we can read. So there's certain signals that we can read in the biosynthetic gene cluster where we can predict what the function of it is. For example, in that case, it's siderophore, and then we can predict, okay, siderophores are usually produced under iron limiting conditions. So then it's one way to actually get the compound to produce. Another way is we can look for the regulators on transcription finding factors, within these gene clusters, and then use molecular biology to actually, enter different regulators or put another promoter in front of certain gene clusters that we can see. These are usually the ways that we try to predict what we can do, how these compounds are regulated. So we're not there yet to do that on a global level, to say, okay, we can easily just automatically detect all gene clusters and know how they're regulated by case to case. In specific cases, we can predict how they're regulated and make certain conditions where they then are produced.
And that would be the genome mining. So the actual production would be the genome mining, or the bioinformatical analysis. What falls under this term, genome mining? That's also in your title of your professorship.
That's actually, I think that's people use genome mining for the whole pipeline. So in my case, it's mostly the beginning. So we really develop tools, computational tools. We develop new algorithms. We develop new ways to predict the activity of gene clusters, to find these gene clusters. So this whole first part. But I think a lot of people also use genome mining, and we do that too, to then you have a certain gene cluster where you do the prediction with the tools that are available, and then you actually get the compound by, for example, heterologous production in a different host organism or so. So I think there's no clear definition. I use it more for the first part of the pipeline, where we detect the genes and we predict things. But I think that there's also people out there that use it for then actually looking for the genome and then using that information to actually get to the compound.
Mm-hmm. Interesting. You've talked about, I believe it was April 2024 episode of the Exzellenz Erklärt podcast.
Yeah.
You were talking about the threat of an antibiotics crisis. What did you mean by that, and how does your research relate to that?
Yeah, that's always very, very shocking to me how few people know about the upcoming antibiotic crisis. So I think everyone knows kind of that there is a problem with antibiotic resistance, right? That a lot of pathogens become resistant to certain antibiotics because of there's too much usage, and then they gain these resistances, and then the antibiotics doesn't work in certain pathogens anymore. Mm-hmm. But I think for not a lot of people, it is clear how bad the situation actually is. So if you look at publications and studies that really showed that by 2050, if we go on like that, we won't have a lot of antibiotics at work anymore, and there will be 50 billion people dying each year of antibiotic resistant pathogens. So infections will be the number one cause of death again.
And this is 2050, yeah. So let's just say 25 years from now. That's what the predictions
are. It's not that far, right? And if you know that usually on average an antibiotic needs 20 years to get on the market, then we actually have to react now and find novel antibiotics. If you think about what kind of wonder weapons these antibiotics are, right? Like you have an infection, you take it, and then after one dose, mostly it is so effective already, and you're healed. I mean, you have to take the antibiotic more, there might be some side effects, but they're so great drugs. And I think most people don't understand the impact or what we lose if we lose the antibiotics, right? And the real function of antibiotics. I think that's scary to me. Yeah.
Yeah, it's scary. I thought I knew about it, but I have to say I'm a little shocked too, because I wasn't aware of that timeline that clearly, as you put it. I think you might have said 50 billion, but 50 million people dying per year.
Yeah, maybe. Yeah, 50. I'm not. It's such a huge number that it's. So it will be the leading cause of death again. So I think and some people talk about a post-antibiotic era, which sounds a little bit like panic. I don't want to create that, but I think just being aware of it and starting to work against that and really finding novel antibiotics with novel mode of action is really what drives my research, I think.
Yeah, I think that's a great point. That's also why I want to go a little more in depth on that point too, because I mean we're looking at kind of a compound problem here, right? We have on the one hand that the increased resistancy, and like you said, the timeline. If we're talking 25 years and the average development of a new antibiotic is 20 years, we have maybe 4 to 5 years buffer to come up with new antibiotics, or we need to be faster or reduce the cost of this research so we can get antibiotics onto the market quicker. So how does what you do, the techniques you use, the tools you use, how does that contribute to either of those two parts of the compound problem?
Yeah, like I said, the way that antibiotics were found in the past was just by looking chemically at these bacteria. You grow them, you extract them, and you find the antibiotic. And one of the big problems was that you tend to find the same antibiotics over and over again, or little changes with penicillin. Instead of penicillin, you develop or you discover penicillin A or penicillin B. But at the end, it's all the same compounds with the same mode of actions, which we don't need really. We really need novel compounds with novel mode of actions to overcome these resistant pathogens. And there was a time a while ago when people were like, okay, maybe we found everything that bacteria can produce already. We have so many nice antibiotics. their work. And so maybe that's all there is out there. And I think the genome mining, or through genome mining now, we are actually able to see what bacteria can produce, or what more bacteria can actually produce. So we just recently published some numbers where we really looked at all the bacteria that have been sequenced today. And we looked at all the secondary metabolite gene clusters that they contain, at least the ones we know. I think, or I believe, that there's more that we don't know how these compounds are made yet. But if we just go based on what we know that bacteria can produce and what is already, what we can see in the bacterial genomes, we know that there's only less than 10%, I think the number was 3% or so of compounds that can be produced by bacteria actually known. So there's this huge potential within bacterial genomes that we have no idea what they actually produce. So there's this hidden treasure really within bacteria that we can go for and find. And of course, not all of them will be antibiotics, right? But there is a huge diversity there that we now have to develop tools for, predict better what kind of bioactivity they have. For example, how do we know which ones go more for or encode for antibiotics instead of anti-cancer or these kind of other activities? And that's where our research goes. So we try to use all this huge genomic data that are out there and predict, based on this data, for example, where can we find more new antibiotics, and try to develop tools how predict better based on the genes which ones are actually antibiotic that encode novel mode of actions or so.
Interesting. And do you have, so how are you already at the point where you can share with the research community, or potentially even like the pharmaceutical market, some of your results where they can start working on that? Because we know the timer is ticking, where they can start going down those avenues, those new avenues that you're opening.
Yeah, I think a lot of people do that already. And I'm not the only one who's developing tools there, right? I think during the last 10 years, just there has been a lot of progress done. I think we now know, for example, in the past, we already looked only at these actinobacteria that produce a lot of antibiotics. But now we know that also a lot of other bacteria can produce antibiotics. And actually, we published a certain list of strains that might, or species or genera that might be worth looking at, that no one looked so far, but we saw in their genomes a lot of potential. And we're also developing tools that predict bioactivity. So it's called target-directed genome mining. That was a method that was developed by other people, particularly Brett Moore in San Diego. But we were able to automate that method so that you can use it for like big data, right?
Nice.
And this method is interesting because it's based on the self-resistance of bacteria to their own antibiotic. So every bacterium that is producing an antibiotic has to be resistant against its own antibiotic, right? To not commit suicide. That would not be very helpful. So, and if we know certain resistance mechanism, we can actually predict what the mode of action of that encoded antibiotic is without knowing anything about the encoded structure of that compound. So that is one of the methods that we use, and we build a tool that's really easy to use for wet lab people. And that's really one of our commitments or our goals, that we make computational tools that are really based on the best methods we have available so far, and that every wet lab person that's working in the lab, every chemist can really easily use without knowing a lot of bioinformatics or something. So really, it should be very intuitive to use, and the output should be in a way that everyone can use it, and that it's really helpful for people not working on the computer.
I mean, that just, first of all, awesome. Second of all, that just begs the question of if someone's listening now and has just heard about this for the first time, where can they find this tool? Like, how can it be accessed as a person working in a wet lab?
Yeah, all our tools are open access and available for everyone. We have a Ziemert Lab page, and we have on my webpage, there's also a list of all these tools, and they're all published as well. And as I said, there's really a great community. So it's not only me in the community, but there's actually a lot of people right now that work together really, that develop tools and databases that really is usable for everyone in the field to really progress in antibiotic research. I think that really is a lot of fun, and that I feel the people, a lot of the researchers in the field are really driven by data now and sharing their results and sharing their tools, so that then other people that are better in the lab work actually can use it and develop or discover novel antibiotics. We do a little bit of that too, but of course, my lab cannot do everything. And I think we're focusing right now on really making these computational tools for other labs.
Mm-hmm. Well, I mean, it's about harnessing the power of the mass, right? Like the mass of data, but also the mass, like just the numbers of researchers who are working on this, and then also the people in practice, and like the translational aspect of actually using it to maybe decrease the time we need for this research or the costs, just so we can find antibiotics or other yet to be discovered helpful.
Yeah, and I think for the data
compounds
we have now, there's also really a lot of potential of using methods like machine learning, right? Some foundation models to develop really different new ways of finding antibiotics. The problem there is that there's still a lack of data, a lack of databases. And I think that's where also we come in as a community to really build new tools and ways and databases that we really have the data, like a lot of data, and a machine readable format that people can use it to develop really good machine learning models for that.
Is that something you're currently doing, or is that something that you plan to do more of?
Yeah, we're doing that too.
Both.
So there's certain things that we can do it, that where we have enough data. But so far, in a lot of cases, it's really still making the databases, which is kind of the annoying part of it. But really making good databases with really solid data to really have good machine learning models for some of the things.
Mm-hmm. Very interesting. Maybe some of our listeners will feel encouraged and inspired to go in on that kind of almost group project. I was also wondering, you said sometime earlier you were referring to soil in some of the examples you used. Given our audienceship, it is interesting to us to also think about what role soil plays in the search for antibiotics.
Yeah, so traditionally soil bacteria were the ones where people looked for antibiotics. So these actinomycetes, or specifically these streptomyces, are very abundant in the soil. And the thought was that the soil microbiome or the soil microbial community is so diverse that there the bacteria really have to fight very hard against each other to be able to grow, because there's so many different bacteria in the soil. And that's why there's a lot of antibiotic producing bacteria in the soil. Sounds good to me, but in general, we think that soil is really fascinating because it's so diverse, right? And if we look we do a lot of metagenomes in the soil as well, or metagenomics from soil as well. And the data that we see there, or the things that we see, is fascinating, because if we look in the soil that we analyze in this metagenomic data in the soil for known antibiotics, which that's where they have been isolated from, these bacteria that produce antibiotics, right? But we can barely see the gene clusters that are encoding for known antibiotics. But instead, what we see is that there's almost all the stuff is unknown and has not even been sequenced so far.
Oh, wow.
So that means that the chemistry that bacteria produce, or the chemistry that surrounds us in the soil, most of it, we have no idea what it is. So, and if you know about the role of soil in like plant growth and how important it is, and how important these secondary metabolites are for bacteria. I mean, they're important for us as antibiotics, but also they're really important for the bacteria. They're signaling molecules, they're defending molecules, right? And if you think about a healthy soil microbiome, then these secondary metabolite, the chemistry that bacteria produce, is really crucial. And we have no idea what that is. Like for most of the stuff, the most abundant and the most abundant gene clusters that encode for chemistry in the soil, we have never seen, or even we don't know what they encode. And that's really was very eye-opening for me, that we think we know a lot, we can do so much, but then if you go into the detail, just the chemistry in the soil, we have no idea what these bacteria in there produce.
But is that something you are looking for maybe a little further down the line with your research to put into focus as well?
Yes. So, I mean, not necessarily in finding the specific chemistry, but right now what we're doing, for example, that's a big community project that we are leading, is we're building kind of a BGC atlas, how we call it. So we're looking at what kind of gene clusters that encode for this bacterial chemistry are present all over the world in all kind of metagenomes. And we developed this website where everyone can look then, if they have a gene cluster or a certain compound that they're interested in, where else is that, and what kind of metagenomes or microbiomes can you find similar gene clusters, to really kind of map the at least genetic basis of the chemistry of the world in these metagenomes. And then, for example, we have a much better way to compare the diversity of this chemistry within different soils or within marine environments or in certain microbiomes, to actually get an idea what is the diversity of the chemistry out there and what can we find there.
Fascinating. The BGC Atlas?
Yes, so BGC stands for Biosynthentic Gene Cluster, which usually we use as a short version for these genes that encode for these compounds.
That's amazing. We will definitely link that as well as the tools you mentioned in our show notes, because unfortunately we're already at the end of our time. I feel like we could talk.
That was fast.
Yeah, we could talk for another half hour. Fascinating, super relevant. Nadine, thank you so much for your time, and I hope to have you back sometime to talk more.
Thanks for having me. That was fun.
Thank you for listening in today. Want to learn more? Then check out our show notes and more info on computomics.com. If you have questions or want to propose a next guest, please reach out to us at podcast@computomics.com.